Everybody, welcome everyone. I realise some people are still going to be on leave, but others have had some leave and now are returning, including one of our speakers for today. As I've said, we will have two speakers from the Animal and Plant Health Agency today talking about their work on SARS-CoV-2. So just a reminder about the UK International Coronavirus Network. We run a series of these seminars once a month, and we will be running some more probably on a quarterly basis in the near future. We would like to remind people to register as members of UK-ICN, and you can do that via the website, in addition to accessing the seminar series. So the seminar series is coordinated by each of our co-directors and covers the themes that are the main areas that the UK-ICN is covering in relation to both of our core funders and also the research institutions for which the co-directors work. But it's also opened for suggestions from speakers from all of our membership to cover our global aspects. So please get in contact with Jill and Lisa through the Secretariat email address to put forward suggestions for future seminar topics. Okay. So today's two seminars, as I've said, are going to be delivered by Fabian Lean and also Rebecca Shipley. You can see their titles there on the current slide. So Fabian had worked for the Animal Health Lab in Geelong prior to coming to APHA. He is a veterinary pathologist, has a very strong interest in infectious diseases, and we have been working quite hard whilst he's been at APHA, working on the viral diseases through the portfolio that I look after. So followed by Fabian will be Rebecca Shipley, who has completed a PhD at APHA on lyssaviruses and is going to talk to us today about the diagnostics and surveillance work that's been ongoing at APHA in relation to doing differential diagnosis and competent authority confirmation of suspect cases in animals at the interface with human cases. So I will take my slide down now and I'll ask Fabian to present his presentation. Thank you.
Thanks, Sharon. I'll share my screen.
Okay, mine should have gone, yes.
Right. So
yep,
hopefully you can
great.
see my screen.
Yeah, that's fine.
So cool. Thanks everyone for dialing in this morning. Yep, so my name is Fabian and I'm one of the veterinary pathologists at APHA, and my scope of work mainly revolves around emerging infectious disease within the animal zoonosis virus disease portfolio under Dr. Sharon Brooks. So today I'm just going to talk about quite a lot of anatomy related stuff on ACE2 distribution in animal tissues and the potential implications on how we understand the host susceptibility of various species. So just quick introduction, as yeah, we're still living in the pandemic. So this virus SARS-CoV-2 emerged in late 2019. The reservoir host and origin has not been determined yet, but it's very likely to be animal origin. The last 2 years, we have also seen heard reports from multiple countries, multiple continents with reverse zoonosis, where viruses pass on from humans to animals, such as in mink, companion animals, dogs, cats, large felids, lions, panthers, and so on in zoos. Most of these animals, when they get infected, they tend to not develop clinical signs or just tend to have a very mild clinical disease, such as just a cough, and they recover very quickly. But as we have seen with the minks in the fur production industries in quite a few countries, these animals develop very severe clinical disease, such as respiratory signs, pneumonia, and unfortunately increased mortality in these production industries. But also recently, we have also heard about stories from the States, from North America, that there have been quite a lot of spillover of SARS-CoV-2 from humans to white-tailed deer. There's current evidence to suggest sustained transmission between deers, and I think there might be a preprint not too long ago that suggests that there might be a case of potential jump of virus from deer back to humans. So I guess there's a lot of implications with policy, animal and human health, but critically, it will also drive evolution of virus as the virus being selected by different immune response, different host species, and so on, as we have seen with the mink in particular. So clinical disease and pathology. So I'm just going to briefly talk about humans, but the main talk of for today will be animals. So in humans, we recognize that SARS-CoV-2 induced COVID-19, which is predominantly respiratory disease, lower respiratory disease, pneumonia, but also there's most of the cases that we get upper respiratory infection, and most of the time asymptomatic or developing a cough. So we have some histology image from autopsies shown on the right here. We have very bad alveolar damage with fibrin deposition, with disruption to the alveolar wall, and quite an interesting feature in humans is a formation of syncytial cells in the alveolar spaces. And in the animal world, the most well reported animal that developed pathology from SARS-CoV-2 infection, natural case, is the mink. So in this case, we've got some pictures from Robert Mollner, a couple of reports from Netherlands, where mink developed very bad pneumonia, as well as probably comparable to humans. There's alveolar damage, there's bronchial damage as well. So it's called bronchointerstitial pneumonia in the mink. So back to basic virology, and looking at the cellular level, SARS-CoV-2, it's mediated for entry through the ACE2 receptor, angiotensin converting enzyme 2. So this allows binding of spike protein and being anchored, and allows internalization to the host cell. In terms of the distribution profile in humans, has been quite well characterized. So we have two human figures here showing the degree of ACE2 expression in tissues. Obviously, if it's rarer, that there'll be more ACE2 proteins being expressed in those organs. And you can see that in particular, the gastrointestinal tract, they express high levels of ACE2. Not very visible here in the human figures, actually at the respiratory tissues. So this links to the diagram below, just to demonstrate the level of expression in on the green on the left, we have the respiratory tract, and on the right, we have gastrointestinal tract. So the larger the circles they are, the higher expression of ACE2 you can find in certain tissues. For example, you can see very big circles in duodenum. This reflects with very red labeling in the human figures here. And as we move down the gastrointestinal tract, the level of laser labeling decrease. On the other hand, in the respiratory tissues, as not a lot of ACE2 expression can be detected. It's mainly detectable within the nasal mucosa and the bronchus, and particularly on the ciliated cells. So I guess in the context of all this disease and infection in animals, do we actually know whereabouts these ACE2 expression are, and could we actually relate to the pathology seen and potentially explain for the difference in susceptibility we have seen with different host species? So what we've tried to do in the last few years is to look for whereabouts the ACE2 receptors are So just one thing to point out, we cannot assume that the ACE2 expression is similar cross species or even a specific taxa. So we really have to examine each species by case. So over here we have 4 species to look at. Here we have ferret, mink, cat, and dog. So this series of pictures have been published previously in a paper describing ACE2 distribution. So I guess the interest with these 4 species, I guess dogs and cats are pretty obvious because of they are companion animals and potentially very high contact with owners in particular. So we looked at whereabouts the receptors are. So for dogs and cats, we don't really see much of a ACE2 labeling in the tissues. Cats, yes, probably up just in the upper respiratory tract, very rare labeling, and some in the submucosal glands in the trachea. But if I could get your attention up to the ferret and mink, just on the 2 rows above, we can see very intense labeling. So when I refer to labeling, they are those brownish red labeling on the mucosal epithelium. And those red arrows should point you to highlight to you whereabouts these receptors are. So if we look at these 2 mustelid species, first we start from the upper respiratory tract, we could see quite a bit of ACE2 labeling in both the respiratory and olfactory mucosa within the nasal cavity. As we move down the respiratory tract, we see decrease of labeling. It's quite a distinct change in ferrets. In particular, we don't see labeling in the trachea epithelium, but we did notice a very weak signal coming out from the submucosal glands. On the other hand, in the mink, we still see the level of intense labeling, the tracheal epithelial and all the way down to the bronchial epithelial as well. So that potentially explains for the difference in host susceptibility. A couple of studies have been published for ferrets and experimentally and mink from natural infection. So on the left, we have a couple of images from one of our recent, not too recent work for ferrets. So we actually developed animal model with ferret to evaluate susceptibility of these animals to SARS-CoV-2. In this particular work, we have only seen immunolabeling of the virus, where we look for the spike protein and nuclear protein, but we also verify such findings with in situ hybridization to look for the expression of spike gene. And we can see very nice correlative images of the expression of proteins and the gene transcripts in these tissues, and they are just focused within the nasal cavity. We've seen labeling in the mucosal epithelial cells. So that could potentially relate to the whereabouts the ACE2 that's shown in previous slides. Whereas on the other hand, mink develop very severe respiratory tissues. We get infection all the way down to the lung. We get infection in the trachea, which can be characterized by necrotizing the tracheitis. We get bronchointerstitial pneumonia in the lung, and this can be correlated with the antigen distribution of the virus, such as seen in figure 11 and 13. So these 2 species is actually quite interesting because even though they are mustelids, they develop a very different clinical outcome. In the ferret, we have not seen any clinical disease or pathology in the lower respiratory tract, whereas as for the mink, we have seen quite severe disease outcome, which can be correlated to the morbidity and mortality reported in the field. So ferret minks are 2 common small mustelids, but there are also other mustelids present elsewhere within the continent and also globally as well. So just want to bring context within what we have in Europe and UK. So in this region, we have large mustelid species such as the European badger and also other smaller mustelid species such as stoat. So in these 2 species over here, we could see very nice labeling. Again, in the upper respiratory tract, very intense labeling, particularly in the stoat. And we see a difference between the European badger and stoat once we progress down the respiratory tract. So we can see with the European badger, we see level of intense labeling in the trachea and all the way down to the bronchial tree. But in the stoat, we have not seen any labeling in the trachea and just very rare labeling in the bronchial tree. So if you compare this side by side with the mink and ferrets, you could see sort of quite distinct difference in the expression over the respiratory tract. And obviously for the European badger and stoat, we don't really know the potential implications in vivo. So that's potential for someone else to be investigating through either surveillance or experimentally. That's another question. We also have interest in other wildlife because of conservation issues and implications as such. So we have also collaborated with our partners in London Zoo. We looked at the Asian short-clawed otter, and we have managed to find ACE2 labeling in the bronchial epithelium as well. Just want to highlight that particularly with wildlife, we tend not to get a full set of respiratory tissues. So most of the time, if we're lucky enough, we just get a piece of lung, and that's the most we can evaluate for, unfortunately. So just back to the positive labeling of the badgers and stoat, there might be potential implications on how we look at infection dynamics in the nature. And recently, there's a group from France that have actually documented evidence of exposure to SARS-CoV-2 in wild mustelids, namely European badgers and pine marten. So just to shift my gear a bit to cervids. So in the early days of the outbreak, various groups have been working on in silico modeling to actually predict for potential host susceptibility in a variety of mammalian species. And this paper in particular by Joanna Damas, she has highlighted a couple of cervids, particularly white-tailed deer and reindeer, that has high binding scores between SARS-CoV-2 spike protein and the cognate ACE2 receptors in these species. And not too long after, probably starting last year, we get news coming out from North America that a couple of white-tailed deer tested positive in the field, and there's also been further evidence, emerging evidence that this deer has also been infected with contemporaneous strains of SARS-CoV-2. This has also been verified by quite a few in vivo studies conducted in the States. And this set of images shown below, it's in situ hybridization looking for the expression of the virus genes. And this set of images published by Matthias Martin showed very nicely that with virus inoculation through upper respiratory tract in the white-tailed deer, the virus could replicate quite rapidly, even just within 2 days post-infection. We get intense expression of these genes for SARS-CoV-2 in the upper respiratory tract, in the lymphoid tissues within the palatine tonsil, but all the way down to the lung as well. So we asked the question if some of the European deer species could potentially be susceptible or potentially be expressing these ACE2 proteins to allow potential susceptibility. So over here, we have a selection of deer species that we have got from various parts of Europe. So we have reindeer, roe deer, and moose. So reindeer in particular was highlighted by Joanna Damas' paper that could be a potentially susceptible host. So we look at the ACE2 receptors, and at this stage, we have not seen any ACE2 labeling in the deer tissues from upper to lower respiratory tract. However, for the roe deer and moose, we have noted some very weak labeling in the upper respiratory tract, and particularly in roe deer, we have noticed labeling the lower respiratory tract, particularly down to the bronchial tree. Just to give a bit of context in terms of the deer species, so roe deer is quite a closely related species to white-tailed deer, so the potential have some implications on how we view the susceptibility of this host, but we're still waiting for emerging data to be able to make that interpretation. Other species that is also present in the continent and UK were fallow deer, muntjac. Muntjac's introduced species from Asia and the late 19th, 20th century, and we have red deer as well, another native species. In these three species, we have only got the lower respiratory tract, i.e. the lungs. So nevertheless, we have seen labeling in the bronchial tree, the conducting airways, and down to the alveoli within type 1 pneumocytes. So at this stage, potentially, if they have been exposed to the virus, there might be likelihood of virus binding to these receptors. But there's also a question of how well do we actually know the ACE2 profiles of different deer species at the molecular level. Camelids thought will be interesting in light of MERS coronavirus. So just a limited subset of species that we look at. Alpacas, we got the whole respiratory tract, and particularly in alpaca, we've seen very weak labeling in the upper respiratory tract, and we did not find any labeling in the trachea or the lung. And as for the bacterium camel, we're quite limited to just the lung, and we have seen very, very weak labeling in the type 1 pneumocyte within the lung. So we also asked the questions if other aquatic mammals or semi-aquatic mammals might be susceptible to SARS-CoV-2. So I think it was last year as well that a couple of hippo that has been tested positive in a zoo in Belgium. So we're quite fortunate that we got some clean tissues from our local collaborator in London. So with hippo, we have seen very patchy, but it's very distinct and definite labeling in the bronchial epithelium. And we have not detected any real labeling in the alveoli. These are just non-specific labeling and does not outline any specific cell population. So this is a negative labeling. And as for seals, also in the backdrop of their susceptibilities to some of the respiratory pathogens, for example, influenza viruses, we're also interested if they could be a potential host for ACE2 mediated viral infection. So over here we have two images from the hooded seal. We have seen very distinct, quite sparse labeling, but it's there within the bronchial epithelium. And deeper down the lung, we have not identified any positive cells in the alveoli. This will be last slide of my ACE2 characterization. So intestines, very briefly, we have screened for ACE2 receptors in all species I've described in the last few slides, and they show pretty much similar expression profile. They're pretty intense on the microvilli of the small intestines, and we've seen quite consistent profile across mammalian species of different taxa. So just to wrap up, in terms of how we understand the anatomical distribution of ACE2, obviously there's high variability of whereabouts these receptors are and the abundance as well across mammalian species down the respiratory tract. So over here with the heat map, we could see difference of labeling, in particular the mustelids, very high labeling consistently in the nasal turbinates across small and large mustelids. And in particular, Ming and Badgers, we could see lots of labeling all the way down to the bronchial tree. And I guess for the Ming, it's quite obvious that the distribution of ACE2 could be related to the pathology seen with those natural infection in the field. As for cervids, we have a couple of species that we have looked at. Generally, they express very low levels of ACE2, nasal turbinate, sometimes in the lower respiratory tract. For felids, we have seen consistent labeling in the nasal turbinates, potentially explain why cats and lions are quite susceptible, but particularly for lions, they develop tracheitis and bronchitis as reported from Bronx Zoo. Other species such as the dog, they tend not to express lots of ACE2 labeling, potentially explain why they are not very susceptible. Pigs and wild boars, that's quite of interest initially in the outbreak because potential concern for disruption to the production and trade. And we have looked at both pig and wild boars. Unfortunately, I don't have the time to show you the images, but essentially I've not be able to identify any ACE2 expression from the upper to lower respiratory tract. And the last species on the right is the alpaca. We have already got some evidence of ACE2 expression, so it'll be interesting to see any comparative studies in the future to look at old and new world camelids because of implications for other coronaviruses as well. Currently, we have preliminary data to suggest some of the pinnipeds, such as the hooded seals shown previously, to express low level of ACE2 in the lower respiratory tract. So it'll be interesting to see what we could get in the future in terms of respiratory tissues, upper and lower respiratory tract, and be able to characterize them fully to be able to make that necessary assessment. So I guess the importance of all these ACE2 labeling work from an anatomical point of view is we could actually be able to advise and link the different receptor usage profile of SARS-CoV-2 variants and SARS-like virus, has been reported by various groups such as in Purbright, and we are aware that, like, with virus variants would have switched tropism for certain species, such as shifting across to rodents, for example. So if we have a baseline understanding of whereabouts these ACE2 are being expressed physiologically in these animals, we might be able to make a stronger judgment on potential host implication. Next point is that if we understood whereabouts these ACE2s are, we can actually target the surveillance on wildlife or companion species much better to understand ACE2-mediated virus infection. And last but not least, but it's fairly open-ended, as we have looked at both domesticated and wild animals. Potentially, we might be able to identify novel animal models that might be useful to understand the pathogenesis of SARS-CoV-2 and SARS-CoV-3 in the future. And last but not least, I'd like to thank the collaborators and funders for all this work. And it's I'm grateful that I've got teachers all the way from the Arctic Circle reindeers all the way down to what we got in UK as well with badges. So thanks everyone for the contributions.